Alzheimer's targets, mechanism of APP cleavage and failures of BACE-1 inhibition. One of the most defining features of Alzheimer's disease is aggregations of the beta amyloid protein in the brain. For over a decade, the dominating hypothesis has been that these aggregates cause Alzheimer's disease by driving tau phosphorylation, tangle formation, synaptic loss, neuronal death, and ultimately cognitive impairment. The formation of these aggregates begins with the cleavage of the amyloid precursor protein into beta amyloid monomers. Because of this, vast amounts of funding and scientific research have been devoted to developing BACE inhibitors, a class of drugs designed to prevent the cleavage of the amyloid precursor protein. Very recently, these drugs have mostly failed in clinical trials. In order to understand the meaning of these failures and their implications for future Alzheimer's research, it is important to understand the mechanism of APP cleavage and of BACE inhibition. Amyloid precursor protein is a transmembrane protein containing the amyloid beta domain. It is expressed at neuronal synapses, where it is thought to play a role in synapse formation and neuroplasticity. Various enzymes, called secretases, sequentially cleave the amyloid precursor protein, releasing various fragments into the extracellular space, as well as into the cell. Alpha, beta, and gamma secretases are all specialized to catalyze cleavage at different sites on APP, resulting in the release of different fragments. In the non-amyloidogenic pathway, alpha secretase first cleaves at a transmembrane site located within the AB domain. It releases soluble amyloid precursor protein alpha, SAPPA, into the extracellular space. Next, gamma secretase cleaves the remaining carboxy terminal fragment, releasing P3 and ACID50. Pathway, beta secretase makes the first cleavage, releasing SAPPB. Gamma secretase then cleaves at its usual site, which in this case releases the entire AB domain into the extracellular space. While the functions of the other fragments are largely unknown, the amyloid beta fragment will go on to aggregate and form oligomers outside the cell, implicating its neuropathological role in Alzheimer's disease. Thus, it is the activity of beta secretase in the crucial first step that allows for the secretion of the amyloid beta domain. The promise of targeting BACE-1, or beta secretase, lies in its aspartyl protease nature, as it has two aspartic acid residues at its active site and a large beta hairpin loop structure that protects this large active site. This ease of binding to the active site of BACE-1 has made it a good candidate for the development of drugs targeting its production. However, BACE-1 also has its limitations, which include difficulty in finding a drug that can cross the blood-brain barrier. Additionally, the large active site and surrounding pockets also mean that the inhibitor must be large enough to bind to the active site, but small enough to maintain the properties of a drug, a challenging feat. Lastly, BACE-1 is required for other cell physiological roles and cognitive function, and inhibiting its functions could prove to be toxic. Though many BACE-1 inhibitors have been developed, the most prominent one is verubicistat. Sarah Vannin et al. 2018 characterized the structure, binding affinity, and stability of the verubicistat molecule in the active site of BACE-1. They found that verubicistat's aminothiodiazine dioxide core uses intramolecular hydrogen bonding of the pyridine and amide groups to bind to the polar and charged residues in the active site of BACE-1. They also found that there is a favorable free energy of binding associated with a BACE-1 verubicistat complex. Despite the excitement and promise of BACE inhibitors, development of 3 BACE inhibitors were recently discontinued in 2018 due to major clinical setbacks. Verubicistat was successful in suppressing human beta amyloid secretion by up to 94%, but was also shown to actually accelerate cognitive decline. In light of these failures, as well as failures of other amyloid beta clearing drugs, some researchers are beginning to abandon the amyloid cascade hypothesis that has prevailed for so long and look for alternative causes for Alzheimer's disease. Recent research has found that soluble amyloid beta serves a variety of physiological roles in the brain. This might explain why BACE inhibitors, whose mechanism stops the production of AB altogether, result in cognitive decline. Some researchers are positing that the increase in beta amyloid production in Alzheimer's is a response to neuronal damage, not a cause. The production of amyloid beta may represent a chronic attempt to protect neurofunctioning. Some researchers are now beginning to shift focus to try to find an alternative underlying cause to this devastating disease.